So welcome everyone. We have the great pleasure to welcome Alice Puppin today, who is going to bring us to the coastal lagoon of the Veneto region and introduce us to this very complex and extremely important ecosystem. I am accompanied by Aditi Dave, who is going to be the co-host of that session. So this is now, we are well into the fall session of Landscapes Live. And you will find all of the necessary information on our website. In particular, we have, I don't know if many of you have seen already, but we have now a Google Calendar that you can add to your calendars where the Zoom links should be found, trying to make your life just a bit easier. And that's about it when it comes to the organization here. So Alice Puppin comes to us from the Veneto region. She finished her master's in environmental sciences at the University of Ca' Foscari in Venice in 2017, and she moved on to the University of Padova for a PhD, which she finished in February of this year. And the work that she will present to us is largely what was done during the PhD, working in the lagoon of Venice in the continued work. Now Alice is a postdoc at the University of Padova. And Alice, let's start. I'll let you share your screen. Thank you very much for accepting the invitation. And we will continue the discussion after the talk. The chat is going to be locked for the moment, and we will open it about 10 minutes before the end of Alice's presentation, and we'll be taking care of reading out loud your questions. Alice, the floor is yours.
Thank you. Yeah, so good afternoon, everyone, and thank you for being here. First of all, I'd like to thank the Landscapes Live team for the invitation to give a talk to this great seminar series. And to be honest, I'm a little bit nervous about living up to the standards. So I hope you will enjoy my presentation, which try to provide an overview of the research I've undertaken throughout my PhD. The subject of the talk will be the role of soil organic matter and carbon in coastal wetlands, and in particular in salt marshes. I will start from a definition of soil organic matter and carbon and an introduction on salt marshes as valuable yet endangered ecosystems, giving an overview of the processes driving their evolution, which importantly involve organic matter dynamics. This will lead me to introduce the three main research questions guiding the project, and then to present how we try to answer to those questions and the major results we obtained. So soil organic matter is defined as the organic component of the soil, consisting of plant or animal tissues in various stages of breakdown, from intact or original tissues of plants and animals to the substantially decomposed materials. Plant residues, the most common, contain 60 to 90% moisture, while the remaining dry matter consists primarily of carbon, which form soil organic carbon, then oxygen, hydrogen, and small amounts of nutrients. Soil organic matter so is important as a nutrient fund, and as it improves soil structure and minimizes erosion. Furthermore, soil organic matter play a crucial role in the carbon cycle, globally containing about 3 times the amount of carbon in vegetation and twice the amount in the atmosphere. As I will elaborate shortly, the role of soil organic matter is even more critical in coastal wetlands. Among coastal wetlands, my research focuses on salt marshes. So what are salt marshes? Salt marshes are intertidal coastal ecosystems characterized by grasslands or dwarf brushwoods on periodically flooded sedimentary platforms. They are covered by the water at high tide and exposed to the air at low tide, and the plants growing on them are adapted to tolerate submersion by saline water. Salt marshes develop worldwide along low-energy temperate coastlines, and being coastal regions strategic points of human settlement, salt marshes are critical yet endangered ecosystems. They provide numerous important ecological functions and deliver a wide range of ecosystem services that contribute to human well-being. They enhance biodiversity, provide nutrient habitats, help maintaining commercial fisheries, protect coastal regions against erosion and storms, filter nutrient and pollutants, and support tourism and recreation activities. In addition, salt marshes, together with other vegetated coastal ecosystems, have been increasingly recognized for their carbon sequestration and storage potential, which make them valuable allies in climate mitigation strategies. Though the future of these intertidal environments is today at risk, exposed as they are to possibly irreversible transitions and transformations due to the effects of climate change and human interferences. So much that between 25 and 50% of salt marsh extent have been lost with respect to their global historical coverage, and they are declining at rates 4 times faster than rainforests. Major threats to salt marsh survival are considered to be direct land reclamation, reduced sediment supply, and increasing rates of relative sea level rise. The high economical and ecological value of salt marshes, along with their fragility, fuel the interest in the preservation and restoration of these environments, and consequently the need to better understand their dynamics. As part of the complex intertidal systems, which is motivated by tide periodic fluctuations, salt marshes are intrinsically dynamic environments, and their evolution is driven by mutual interactions and adjustments among hydrodynamic, morphological, and biological processes. Being a great example of ecomorphodynamics. Schematically, water flow over marsh surface drives sediment transport, deposition, or mobilization, shaping marsh morphology, which in turn influences hydrodynamics. Allophytic plants, spatially organized in characteristic patches related to soil elevation and flood frequency and duration, has a key role in marsh evolution, influencing the water flow, enhancing sediment settling and stabilization, and producing biomass that contribute to soil formation. Thus, sediment deposition and organic matter accumulation together contribute to the peculiar land building capability of salt marshes, which allow them to keep pace with relative sea level rise. Although organic matter content is strongly variable in space, in some conditions, soil organic matter may contribute vertical accretion up to 1.5 to 3 times more than inorganic matter, according to Allen and others, 2021, having a key role in the survival of coastal wetlands with low mineral supply. Enhanced knowledge in organic matter dynamics would benefit the models, trying to reproduce the physical and ecological processes driving marsh evolution. In this process, thanks to primary production coupled with surface accretion, soil organic matter accumulation allows salt marshes to capture and store carbon, removing CO2 from the atmosphere through plant photosynthesis and by trapping organic debris within the soil. Some estimates show that the carbon accumulation rate in vegetated coastal ecosystems, including salt marshes, but also mangroves and seagrass meadows, may be exceptionally high, up to 1 to 2 orders of magnitude higher than in terrestrial forests. So in this framework, soil organic matter has an important role within salt marsh environments, as on one hand, it contributes to the building up of marsh elevation, thus to marsh survival, and on the other hand, it supports the carbon sink potential of wetlands. And however, the effects of climate change and human pressure may alter the ecomorphodynamic feedbacks driving marsh evolution, thus causing salt marsh drowning and the loss of related ecosystem services. For this reason, there is a growing interest toward organic matter dynamics in salt marsh environments, and this motivated our research. Soil organic matter in marshes is constituted by a combination of in situ production of below ground biomass, for example, roots, rhizomes, and tuber tissues inserted into the sediments, and autochthonous or alloctonous organic materials that are deposited on the salt marsh by tidal currents. Then the fate of soil organic matter strongly depends on outputs, which are mainly caused by decomposition, which determine the fraction of organic matter produced or imported that effectively contributes to soil formation and carbon burial. Inputs and outputs, and consequently, of course, organic matter variability, are affected by numerous intertwined factors, often acting simultaneously and occurring over a wide range of spatial and temporal scales, including, for example, climate, vegetation characteristics, sediment properties, and geomorphology. So, toward the goal of improving our understanding of organic matter dynamics influencing coastal wetlands resilience and its carbon sink potential, our project focused on 3 main research questions. The first, how decomposition affects soil organic matter and which are its physical and biological drivers. The second, which are the spatial and vertical patterns of soil organic matter and which are the physical and biological drivers of soil organic matter variability. And third, what are the dimensions of carbon stock and carbon accumulation rate at our study site? The study site selected for our research is the Venice Lagoon in the northwestern Adriatic Sea in Italy, being the largest lagoon in the Mediterranean. The Venice Lagoon is a back barrier system connected to the sea by 3 inlets. And the Venice Lagoon is a shallow microtidal basin characterized by a semi-diurnal tidal regime with an average tidal range of 1 meter and mean water depth of about 1.5 meters. It is an area of wetlands, marshes, navigable channels, and islands, hosting the famous city of Venice, listed as a World Heritage Site. So the Venice Lagoon is a paradigmatic case for comprehending anthropogenic and natural forcing interactions, as here coastal populations and wetland environment have been intertwined for centuries. The present-day morphology of the Venice Lagoon, which formed during the Holocene transgression, is the result of a combination of natural forcings and human interventions. In fact, since the 15th century, to prevent the infill of the lagoon and preserve channel and port functions, several hydraulic works have been carried out, including river diversions, canal digging, and inlet modifications, which strongly modified sediment supply and local hydrodynamics, favoring sediment starvation and salt marsh loss. As you can see from these maps, the salt marsh areas represented here in green have significantly decreased, going from about 200 square kilometer in the 16th century to less than 50 square kilometer today. Finally, in October 2020, some storm surge barrier known as the Moses system became operational to protect the city of Venice from flooding, and we will talk about them later. So, starting with the first research question, I'm going to present our analysis on decomposition in salt marsh soils. What is decomposition? Decomposition is the process of decaying that organic matter, which is broken into simple compounds through fragmentation, leaching of water-soluble compounds, and microbial catabolism with the release of CO2. Decomposition is controlled by the interactions between physical environment, quality of the substrate, and community of decomposer organisms. To analyze decomposition processes in marsh soils, we used the TBAG index protocol proposed by Consequently and others in 2013, which consists of a simplified leaderback experiment with standard litter, which enable to investigate the influence of environmental conditions. The standard litter consists of two types of commercially available tea bags with different properties in terms of resistance to decomposition. Rooibos tea, more recalcitrant, and green tea, more labile. Pairs of tea bags were buried in the soils of 7 salt marshes for 90 days, and the remaining mass of tea was measured after retrieval. The TBAG index allowed us to measure decomposition rate and litter stabilization factor and their relationship with some environmental variables. Environmental conditions which we considered are water temperature, marsh elevation, distance from the marsh edge, and flooding percentage. I will not go into the details of the method, but what is important to know is that decomposition rate tells us, of course, how fast decomposition proceeds, and litter stabilization factor indicates the fraction of labile material that becomes stabilized during the process, chemical properties of the tea being known. The use of 2 tea types allowed us to obtain the 2 parameters during one single incubation period. As to the results, we found that the mean value of the decomposition rate calculated for the salt marshes of the Venice Lagoon is 0.012 days minus 1. And this means a loss of about 70% of the initial labile mass after 90 days, which is quite a lot. However, if compared with previous findings from other environments, it confirms salt marshes as some of the biomes with slowest decomposition. Our results confirm also that increasing temperature may accelerate decomposition. suggesting an exponential increase of organic matter decomposition rates with temperature and a linear decrease of stabilization factor with temperature from about 19% to 12%. Furthermore, we observed a statistically significant relationship between the decomposition rate and both the marsh elevation and the distance from the marsh edge, with decomposition increasing with elevation and decreasing moving farther from the marsh edge. Considering the morphological characteristics of the marshes we analyzed, this suggests that oxygen availability influenced by the interplay between marsh morphology and water levels plays a significant role in wetland decomposition and affects its spatial variability. However, the results on flooding were not what we expected. No significant relationship with K and S decreasing with increasing flooding percentage. One possible explanation for this result might lie in the method used to calculate the flooding percentage, as it was indirectly calculated by comparing site-specific measurements of marsh elevation with tide level data from nearby tidal gauge stations. Therefore, these might not be an optimal representation of the actual presence of water above the soil, as it may depend also on local marsh microtopography, for example, or on tidal creek networks. Finally, we observed wide differences in decomposition of gritty, representing labile material, and rooibos tea, representing recalcitrant litter. And this highlights that the initial litter quality, for instance, if there are more or less wood tissues that are more resistant to decomposition, may have a critical role in determining organic matter decay and carbon accumulation. And this gives weight to vegetation type and its distribution. If you were interested in obtaining more information on the topic, you can find it in the paper that we've recently published in JGR Biogeosciences. So, moving to the second part of our research, I will now focus on soil organic matter variability and distribution patterns. Our investigation on organic matter and carbon content based on the analysis of 720 soil samples from 60 sediment cores to the depth of 1 meter. 1 meter is usually the reference layer in soil organic carbon estimates, and that's the reason why we selected this depth. The cores were collected along linear transects from the marsh edge to the inner part in 10 different areas of the Venice Lagoon to capture the variability of soil organic matter both at the marsh scale and at the lagoon scale. Core collection points were surveyed using a GPS, and vegetation characteristics were recorded as well. Soil samples were taken at 12 depths from each core, and they were analyzed for soil density, sediment grain size, organic matter content, and carbon content. To measure organic matter content, we used loss on ignition, a relatively cheap and rapid method that estimates organic matter based on the change in weight after the combustion of organic matter in a muffle furnace. To estimate the soil organic carbon, we directly measured the organic carbon content using elemental analysis on a subsample, and then we calculated a conversion equation from LOI values to carbon content. As you can see, Organic matter content observed showed a wide variability, vertically, spatially, both at the marsh scale and at the lagoon scale. So what influences this variability? First, we considered sediment characteristics, and we observed that organic content shows a decreasing trend with coarsening sediment grain size. A possible explanation for this relation may be that fine sediments have a greater potential for organic mineral interaction and aggregates due to their greater specific surface area. In addition, fine sediments are known to provide better preservation conditions from decay, being characterized by lower oxygen exchange. Then, to inspect present-day drivers of soil organic matter dynamics, we focused on the top 20 centimeter soil layer, as it is the layer directly impacted by current vegetation. And of course, we observed that the vegetation had an impact. Organic matter content significantly increases with species richness, and the presence of some species with abundant and recalcitrant biomass may favor higher organic content, such as in the case of Limonium narbonense, characterized by massive woody roots, and Phragmites australis, a reed plant growing close to freshwater inputs with high above ground and below ground biomass and quite recalcitrant tissues, as we found them well preserved in quite old sediments. At the marsh scale, we observed that organic matter content generally increases with the distance from the marsh edge. At the lagoon scale, organic matter variability is guided by the gradient generated by marine and mainland or fluvial influence, with higher organic matter content found toward the mainland. So variations in sediment type, vegetation, morphological and hydrological dynamics affect both organic inputs and preservation at different scales. At the marsh scale, the enriched organic matter content of inner marsh may be due to the preferential deposition of denser inorganic sediments on the marsh edge and transport of less dense organic particles toward the inner marsh during tidal inundation. Furthermore, inner, often lower marsh areas may be characterized by frequently flooded anaerobic soils where decomposition is slowed down, as shown before. At the lagoon scale, freshwater inputs promote the growth of high primary producer reeds and may provide organic rich sediments. In addition, inner areas were observed to be characterized by finer sediments due to the gradient of hydrodynamic energy, which, as we've just discussed, may be characterized by higher organic content. As to the vertical variability, depth distribution of soil organic matter is often assumed to be exponential, and this assumption is used to predict organic content at unsampled soil depth. However, we observed highly variable depth distributions, rapidly decreasing with depth at some sites, but also showing peaks at different depths at other sites. And we thought this was probably related to the variation of environmental and depositional conditions through the time. So, to better understand the observed variability, sedimentological analyses were carried out on the study course in order to link sediment characteristics to the intertidal deposition environment. Through the observation of color, grain size, texture, sediment structure, and biogenic content, we were able to distinguish 4 depositional subenvironments. Salt marsh deposits, formed by the accretion of current salt marsh environments with abundant live roots and plant debris, sometimes concentrated in visible laminations. A so-called brackish marsh deposit, distinguished where reed fragments were recognized, testifying current or past near freshwater or palustrine conditions. Then tidal flat deposits, defined by the absence of roots in live position, poor plant debris, sandy laminations or massive coarser sediments. And finally, a transition zone between salt marsh deposits and tidal flat deposits, being characterized by features from both these environments, corresponding to tidal flat deposits colonized by pioneer vegetation. This allowed us to investigate the evolution of our study sites and assign to each sample a depositional subenvironment. As an example, at this site on the top, we observed the formation of a salt marsh above a tidal flat. In contrast, at this site on the bottom, we discovered a brackish marsh deposit with well-preserved reed fragments beneath the existing salt marsh. And the brackish marsh deposits likely date back to a time when the mouth of a river, which was later diverted, was located in this area. Our results show that marsh deposits are characterized by significantly higher organic matter content than tidal flat and transitional deposits. And furthermore, they revealed significantly higher organic matter content in brackish marsh deposits than in salt marsh deposits. These patterns is affected probably by vegetation characteristics, with biomass production decreasing from marsh environments to tidal flats, and the presence of reeds characterizing brackish marsh deposits. Furthermore, it is in accordance to grain size properties characterizing the different environments, with marsh deposits being constituted by finer sediments and tidal flat and transitional deposits by coarser sediments. Soil density and organic carbon content data were used in the third part of our research to estimate the carbon stocks and carbon accumulation rates in the salt marshes of the Venice Lagoon to assess their blue carbon sink potential. The term blue carbon was coined in the early 2000s to indicate the biological carbon captured by marine living organisms as a counterpart of the iconic terrestrial forests called green carbon, to emphasize the fact that over half of the global carbon sequestration is attributable to marine and coastal ecosystems. Global interest in blue carbon is rooted in its potential to offer a nature-based approach to climate mitigation while achieving co-benefits, which are the numerous functions, ecological functions and ecosystem services provided. And importantly, draws attention to the degradation of and the need to conserve and restore coastal environments. Some estimates showed that the carbon sequestration and storage potential in vegetated coastal ecosystems, including salt marshes, may be exceptionally high, as you can see here, comparable or even larger than terrestrial forests. But again, as you can see, these estimates are still affected by high uncertainties. Organic carbon storage measurements include organic carbon stock, which is the quantity of organic carbon contained in a reference soil interval, and organic carbon accumulation rate, which is the quantity of organic carbon added to the soil in a reference time, both relying on carbon density of the soil. Although the depth of marsh deposits may be variable, as we have seen, the reference soil interval in carbon assessment correspond to 1 meter, and we stick with this rule. Carbon stock in our work was calculated by the sum of the carbon content at each soil interval sampled, resulting from the product of soil carbon density and interval thickness. Accordingly to organic matter content variability, which we observed before, carbon stock resulted to be highly variable in different marshes, with higher values generally occurring at the landward side of the lagoon. Our estimates support salt marsh important as a carbon sink environment, being comparable, as we can see here, to carbon stock estimates attributed to temperate and tropical forests. And high content of organic carbon found deep in salt marsh soils with well-preserved plant debris dating back hundreds of years confirms salt marsh potential to sequester carbon over long temporal scales, and thus the eligibility of their carbon offsets for greenhouse gas markets. Furthermore, our results give us an information on the amount of carbon which would be mobilized and potentially lost if salt marsh after salt marsh erosion, degradation, or conservation, or conversion, sorry. Carbon accumulation rates were calculated as the product of soil carbon density within 5 centimeter topsoil layer and marsh accretion rate estimates, both representing recent conditions. Carbon density displayed local mean values ranging between 0.017 and 0.031 gram per cubic centimeter. And marsh accretion rates were derived from measurements based on marker horizons over 2 to 20-year period. In the microtidal sediment-starved system of the Venice Lagoon, accretion rates are of the order of few millimeters per year. Nevertheless, considering the order of magnitude of the two variables in the equation, is evident that accretion plays a strong role in the calculation. Our estimates of carbon accumulation rates ranging from 49 to 113 tons of carbon per square kilometer per year fall within the range of variability defined by previous estimates, although lying in the lower part of the range. We observed that the reason for this position is mainly that higher estimates from previous findings include higher accretion rates from more energetic systems. Our results allowed us to estimate that the salt marshes of the Venice Lagoon, which cover an area of about 43 square kilometers, may sequester over 13,000 tons of CO2 per year, which is equal to about 20% of annual emissions from the waterborne navigation occurring in the city of Venice, or in other words, more than the emissions from 200 cars. This confirms the role of salt marsh ecosystems in contributing to mitigating greenhouse gas effects. And these contributions has a monetary value, which can be assessed in carbon offset markets. Based on the average spot price of European emission allowances in 2022, the value of the annual CO2 sequestration estimate from the salt marshes of the Venice Lagoon is about 1 million euros per year. At this point, we wondered what the impact of flood regulation strategies, such as the Moses system, could be on marsh carbon sink potential. We've just mentioned the critical role of accretion rate for carbon accumulation, and Tonyin and others in 2021 observed that the operation of the storm surge barriers of the Moses system further reduces salt marsh vertical accretion, as they reduce water level within the lagoon during storm surges, which are fundamental suppliers of sediments to salt marshes. They estimated a vertical reduction on marsh platforms of 1.1 millimeter per year. Hence, in the current flooding regulated scenario, we observe a reduction in carbon accumulation rates ranging between 25 and 48%, with a general reduction of the CO2 sequestration potential of 33%. With this, I'm coming to the conclusions, and to sum up, I'd like to summarize the main outcomes of our research as follows. Decomposition has a crucial role in determining the amount of organic matter that effectively contribute to soil formation and carbon accumulation. While salt marshes exhibit lower decomposition rates compared to other environments, labile materials still decay rapidly. However, the rate of decay varies accordingly to local conditions, temperature, morpho-hydrological factors, and organic litter quality are key factors in decomposition processes. Those changes in climate, hydrodynamics, and vegetation may critically affect organic matter preservation. Then our research has highlighted the substantial variability of soil organic matter at the lagoon scale, both spatially and in the vertical dimension. This variability should be considered when assessing organic matter and carbon content in marsh environments. Inputs and preservation of soil organic matter and carbon are importantly affected by sediment type, vegetation characteristics, and morpho-hydrodynamics, which differ across different geomorphic settings and depositional environments in space and in time, and this claim for site-specific data. Finally, our results shed light on the significant role tidal marshes played in carbon sequestration in the past through the development of organic carbon stocks, and may play in the future through carbon accumulation, although emphasizing a wide variability again. Our findings underscore the significance of salt marshes as blue carbon ecosystems, demonstrating their potential as valuable allies in climate mitigation strategies. However, their survival and the services they provide are importantly affected by the choices made in coastal management. So we believe that enhanced knowledge in this field will empower us to make informed decisions about wetland management and restoration strategies, and only careful coastal management strategies can secure the long-term health and functions of coastal ecosystems, as well as the well-being of coastal population. I would like to conclude by thanking my supervisors, Professor Dalpaus and Marani, and all the collaborators on this project, and the research team that you can see here. And finally, of course, thanking again Landscape Live team and all of you for your attention.
Thank you so much, Alice. It was fantastic.
Thank you.
The last picture you showed us of your team, it was not the lagoon, right? Or I am
No,
okay.
No, it wasn't.
So everyone, the chat is open. You can write down your questions in it. We will start maybe with a few questions from Aditi and myself while you're putting your thoughts together and writing them down. And we have a bit of time ahead of us for discussion. I noted there are quite a few questions as you were talking, and let me just try to see, where do I begin? I suppose that one thing that I was wondering, just to get a sense of the rates of evolution of the system, is you showed us that in 1881 we were still looking at a fairly natural system without the waterworks. How deep is the layer of 1881 in the salt marshes you investigated? Is it something that is just a few centimeters below the surface, or is it already a meter below the surface? I'm lacking the context here.
Okay, yeah. So I would say that that's the depth. I mean, dating of sediments would be quite different in different parts of the lagoon. So accretion rates could be different in different parts of the lagoon, but as a general estimation, we think that in the first meter we can have also 300 years of history, I would say. Yeah, but of course it also, the accretion rates were not constant. And for example, in the southern lagoon, close to the former mouth of rivers that then were diverted, they could be higher as due to the river inputs, and they could be lower in other parts of the lagoon where sediment inputs were lower. So
pretty high variability.
Yeah, but yes. Yeah, I would say, yeah, first meter could be more or less 300 years.
I see that Aditi told me that she has an issue with her webcam that's not working, but I'll let her ask a question now.
Yeah, sorry, my camera is not working, but a really great talk, Alice, by the way, really interesting. So my question might be quite subjective, and it's probably, it depends on, from site to site it varies, but to what degree do you think that the decomposition rate, like the organic matter decomposition, is affected by vegetation type versus all the other factors? I guess you can only speak for your sites, but, just to have an idea.
Yeah, I think this is a very interesting question, and we also thought about this team, and previous findings show that, for example, lignin content in plant tissues could be a good proxy for understanding the resistance to decomposition. So as we observed high difference in decomposition rates between labile and recalcitrant material, I think that vegetation characteristics could be really important. And we, for example, found really well preserved really well preserved litter from reeds that were recognizable. They looked as they were from present day, but they were like 80 centimeters deep. So probably they were there for really a long time. And this is probably due to the high lignin content, I would say, of this type of plants. So I would say that, yeah, this is probably an aspect that would be interesting to investigate. But I think, yes, that definitely vegetation characteristics and species composition could be an important factor.
Oh, okay, yeah, thank you.
Thank you.
I'm going to follow up with a very interesting question from Grace Montoya, who's asking how would you compare or apply the methodology in tropical areas in Colombia, for example? Could you just transpose your approach to any types of climate, or is there some specific elements that are like Mediterranean climate and versus tropical climates?
Is this related to the TBEG index methodology, or
I don't know.
General?
Maybe Grace could write down if it's if there's a specific part of the methodology that that you were thinking about. But let's start with the TBEG index.
Okay.
Could you use it in any type of climate?
Well, I know that TBEG index proposed by Consequently, was used in very different part of the world, and it was designed with standard litter to be comparable in different conditions. So I think it could be used in different climates. So he says same methodology probably in general.
Yeah, I suppose.
Yeah.
But so then I guess the answer is probably yes.
Yeah, I would say that the methodology could be applied in other environments, also in tropical areas. Probably we would have different conditions that we should take into consideration, but I would say that yes, the methodology is pretty standard.
So more important than the climate is whether your supermarket is carrying rooibos tea and green.
Yeah, that could be a problem because the one that we used, I think they are running out of. Yeah, they are not produced anymore probably, but you can find another type.
So I'll as I was about to ask my question, I'm going to read one by Kyung So Yoo. Excellent talk in many ways. Thank you. They're curious what role salts play in the interaction between organic matter and minerals. For example, in terms of absorption of organic mineral on the mineral surfaces of organic matter, occlusion in aggregates. Yeah, so the role of the particular role of salt in that system.
This is interesting. To be honest, I'm not an expert of this. And I think we didn't consider the possible interaction between salts and mineral and organic matter aggregates, but we observed that organic content in different areas of the lagoon with different salinity were different. However, the direction of the relationship was the opposite to what we expected from previous literature findings. So we thought that maybe other factors overcome the influence of salinity. But I think, we could consider and deepen this aspect. Could be interesting.
Thank you. Aditi, would you like to take the next question?
Yeah, I could do that. So there's the question by Deborah, who's asking whether you consider the effect or anaerobic conditions on the carbon content of the various zones.
So anaerobic conditions, yes, we, for example, in the TBEC index experiment, we tried to consider this aspect as we considered among our environmental conditions some factors that could be considered proxies for aerobic or anaerobic conditions of the soil. So we took into consideration elevation and distance from the marsh edge, considering the typical morphology of marshes we analyzed, and also flooding percentage. But as I said, the results was not so clear, probably due to the methodology used to calculate flooding percentage. But I would say that anaerobic conditions, aerobic or anaerobic conditions, are variable in time in salt marsh environments. So rather than comparing them between zones, I would compare them in different moments, and probably elevation and distance from the marsh edge was the best proxies we was able to get for taking into consideration anaerobic conditions.
All right. Thank you very much. This brings us to the hour. And don't hesitate to reach out to Alice if you have further questions. Her email is on the slide right here. Of course. And thank you, everyone, for participating. Thank you very much, Alice, for taking the time to introduce this topic in this area to a community that is that should learn more about it, I suppose. This was very enjoyable. Thank you very much.
Thank you too again. Thank you, everyone.